DNA extraction methods. What is a DNA? DNA, also known as deoxyribonucleic acid, a fundamental molecule found in all living things, carries the genetic information in the cell, contains instructions for our body cells to perform their specific functions. The sequence of nucleotides determines individual hereditary characteristics. Basic unit of information in DNA is the gene. Human beings have about 25,000 to 30,000 genes. Size of organism's genome is roughly a measure of its complexity. Viruses 5 to 10 kilobits, E. coli 4,640 kilobits, human 2,900,000 kilobits, about 2.9 billion base pairs. DNA extraction. The first isolation of DNA was done in 1869 by Friedrich Miescher. Currently, DNA extraction is a routine procedure used to isolate DNA from the nucleus of cells in molecular biology or forensic analyses. DNA can be extracted from different sources: blood, semen, saliva, urine, hair, teeth, bone tissue. DNA extraction procedures: organic phenylchloroform extraction, non-organic proteinase K and salting out Chelix ion exchange, resin extraction. Organic phenylchloroform extraction, cell lysis buffer, lyse cell membrane, pellet nuclei, resuspend nuclei, add sodium dodecylsulfate SDS, proteinase K, lyse nuclear membrane and digest protein. DNA released into solution is extracted with phenylchloroform to remove proteinaceous material. DNA is precipitated from the aqueous layer by the additional of ice-cold 95% ethanol and salt. Precipitated DNA is washed with 70% ethanol, dried under vacuum and resuspended in Tay buffer. Steps in organic and inorganic DNA extraction: 1. Lysis of red blood cells (RBC). 2. Digestion step: lysis of white blood cells (WBC). 3. Phase separation step: extraction of protein, organic DNA extraction (PCI). Inorganic DNA extraction: 6. Sodium chloride. 4. DNA precipitation. 5. Washing with ice-cold ethanol. 6. Dilute the pellet. (1) Lysis of red blood cells (RBC). Lysis of red blood cells: added 800 ul of tris EDTA buffer, tris HCl10 EDTA 2 in 200 ul ml of the blood. Mixed by inverting several times. Centrifuged at 5,000 revolutions per minute for 10 minutes. Discarded the supernatant. Break the pellet formed at the bottom of the Eppendorf tube by tapping it gently. Add 1 ml Tay buffer and mixed it gently. Centrifuged at 5,000 revolutions per minute for 10 minutes. This step may be repeated until pellet becomes light pink. 2. Digestion step: lysis of white blood cells (WBC). Pellet obtained after lysis of RBCs resuspended in 400 ul buffer TNE, tris HCl10 EDTA 2 and NaCl 400 200 L 10% SDS 50 L proteinase K 50 L of 10 micrograms all conc. Homogenize the tube with gentle rotation. Samples incubated overnight in 58 degrees Celsius in shaker water bath. Next day. Phase separation step. Extraction of protein. In this step, we can remove the digested protein through 6-am sodium chloride inorganic method, or phenylchloroform isoamyl alcohol (PCI), in ratio 25:24:1 respectively. Organic method. DNA released into solution is extracted with PCI to remove proteinaceous materials. Add equal volume of phenylchloroform isoamyl (PCI) alcohol. Mix gently for 2 minutes and centrifuge for 10 minutes at 10,000 revolutions per minute at 4°C. Carefully remove the top aqueous phase containing the DNA using a 1000 ml pipette. Transfer to a new tube. 4. DNA precipitation. Precipitate the DNA with absolute isopropanol and inverted the tubes gently till DNA threads became visible, and then left the tubes at room temperature for 10 minutes. Centrifuged at 8000 revolutions per minute for 10 minutes and discarded the supernatant carefully, and white pellet of DNA made visible at the bottom of the tube. 5. Washing with ice-cold ethanol. Wash DNA pellet with 1 ml of 70 to 100% ethanol. Break and mix the pellet, then centrifuged at 8000 revolutions per minute for 10 minutes and discarded the supernatant carefully. Air dried the DNA pellet at room temperature for at least 2 hours. Organic extraction and inorganic extraction. Advantages: yields relatively pure, high molecular weight DNA. DNA is double-stranded, good for RFLP. Disadvantages: time consuming, requires sample to be transferred to multiple tubes, increases risk of contamination, involves use of hazardous and smelly chemicals. DNA extraction by CHELIX method. Make 5 to 7% solution of Chelix aliquot 300 ml solution in 15 ml Eppendorf tube. Take 300 ml blood and 3 ml distilled water or RBC lysis solution. Centrifuge at 5000 revolutions per minute for 2 minutes. Repeat lysis step. Add 300 ml 5 to 7% Chelix solution. Vortex for 15 to 20 seconds. Place tube in heating block at 95°C for 20 minutes. Vortex for 15 to 20 seconds. Centrifuge at 10,000 revolutions per minute for 2 minutes. Transfer the supernatant in Eppendorf tube and use as Chelix extraction. Advantage is relatively fast, can extract directly from cloth stain, minimizes contamination, uses only a single tube, removes PCR inhibitors. Disadvantages: results in single-stranded DNA, not useful for RFLP. Practical applications of extracted DNA in DNA technology: medical diagnosis, disease, human gene therapy, pharmaceutical, hormone production, insulin, human growth hormone, forensic DNA, fingerprinting, agricultural, plant breeding. Thanks for listening. For more videos, please like, share and subscribe our channel.